teachers, because they will really know what's in the exam questions. The ones with the ticks are the ones where it's actually specified in the syllabus. Those of you where you don't need to know about this level of detail, you just need to understand and learn the content of my photosynthesis video, because that is a simplified version of the light-dependent reaction. So really quick bit of context, let's just say it just in case you didn't know. This is practically the most important energy transfer reaction on the planet, because it transduces light energy into chemical energy. It's the light energy that powers the reaction between carbon dioxide and water to make glucose and oxygen. Glucose being the foundational molecule that plants make every other molecule that they need from. And it is the photosystems, these complexes of proteins and photosynthetic pigments that contain chlorophyll, kind of obviously, that absorb the light to allow this to happen. But I'm going to take you back to a bit of GCSE, because I'm hoping that you'll recognize this, because basically leaves are the solar panels of plants. And if we're going to be really specific, it's kind of actually the photosystems that do the nitty-gritty of that job. Lisa, hopefully we'll get this sorted out. We're still on the introduction. So this is a cross-section of a leaf, which I suspect you'll recognize. And we've got the palisade layer at the top, packed full of chloroplasts. That's where the photosystems are. It's where the chlorophyll is. And the spongy mesophyll layer just below the palisades, which have also got a whole load of chloroplasts with them. Nice little activity to do. Just pick a leaf from any plant, or maybe not even pick it. Just turn it over and you will see that the top of the leaf will always be much darker green than the bottom. That is evidence for the fact that this top palisade layer is the one that is most packed full of chloroplasts, because that's where the chlorophyll is, and chlorophyll is green. We see it as green. It's why plants are green. Okay, oh yeah, that's just an emphasis that light energy makes that happen. So really quick summary of photosynthesis. You'll remember, this will almost be nostalgic for you. This is your GCSE summary equation for photosynthesis. Sadly, you probably know that it is far from this simple. And this reaction takes place in the chloroplasts in the leaves of the plant. And Hamid, not a disgusting topic. No, no, no. I'm hoping you might end up enjoying it by the end of this next half an hour. Okay, we are going to nail this and you are going to understand it, and it isn't going to be quite so disgusting. There is my aim for the end of this half an hour. So chloroplasts, just notice I haven't coloured any of this in green, because the only thing that is green in a chloroplast is the chlorophyll pigments. There you go. So double membrane of the chloroplast, just like the mitochondria does. And then we've got these single membrane, I call them plates. They're thylakoids, and the granum is the stack of plates. And every now and again, you have a plate joining another plate by a bit of membrane called the lamella. Oh yeah, and all the, you know, the stuff that is around the jelly-like matrix, as it were, that is around the granum is called the stroma. Don't be confused. It is called the matrix in the mitochondria, but it's the stroma here. And you probably know that photosynthesis is split between the light-dependent reaction and the light-independent reaction. So don't be fooled. Even though the light-independent reaction doesn't need light for it to happen, it absolutely needs products of the light-dependent reaction for it to occur. This baby is going to stop really, really quickly, and we're talking milliseconds if this one stops. Okay, so light-dependent reaction. Let's get my... So light-dependent reaction. This uses water and light energy to make oxygen. But while it is doing so, put really crudely, don't quote me too much on this, but you can think of this bit as the light dependent reaction. This produces ATP. This is photophosphorylation. Phosphorylation just means adding a phosphate. That's all it is. Photophosphorylation means that you're adding a phosphate using light energy. And when we talk about phosphorylation, we're nearly always, and we're just using it as a term, so you talk about in respiration substrate level phosphorylation and oxidative phosphorylation. Here we're talking about photophosphorylation, and those are the three ways in which ATP is made. Because when we use that term like that, phosphorylation, we're actually talking about the phosphorylation of ADP, the addition of a phosphate to ADP to make ATP. This reaction also makes reduced NADP. This is a coenzyme using an adenine ribose nucleotide as its basis. So just it's kind of linked to your nucleotides, this thing here. And the other thing on the shopping list that has to be made from the light dependent reaction is oxygen. I want you to take note of these three things in blue, because we are going to have to make these three things on our shopping list in blue as we go through, particularly non-cyclic photophosphorylation and cyclic. All right, so just take note of these three things. The light dependent reaction has to make all of them. The oxygen is used in aerobic respiration because, of course, plant cells are aerobically respiring pretty much 24/7. They're certainly respiring 24/7 because they have to produce ATP for all of those biosynthesis of biomolecules. If more photosynthesis is going on than respiration in the cell, which of course it does on a nice sunny day, there is more oxygen produced than is required for respiration. And therefore, during a sunny day, oxygen diffuses out of the leaf via the stomata. What do I do? Oh, no, AQA. Hamid, AQA. No, you don't. You need to know my photosynthesis video. And now you know that you don't need this level of detail. You can go and have a cup of tea. Okay, so yeah, you don't. Sorry, just seen that. Will these be available? Yes, it is. Yes, I keep the live streams on my channel for a period of time after each live stream, at least a few weeks. So, wait, O2 is produced during photolysis. Yes, it is. Yeah, we're going to go through that and explain exactly how that happens. That happens as part of the light-dependent reaction. Hamid, I've just made you very happy. Pleased about that. So you need to know this bit, though, Hamid. Don't sign off quite yet. So the ATP and the reduced NADPH is absolutely required for the light-independent reaction. If these babies aren't made, this ain't happening. For the light-independent reaction to happen, CO2 is fixed to make, if I'm going to be accurate, it's fixed to make triose phosphate, or GALP if you do the syllabuses that use that term. And the triose phosphate goes on to make glucose and other organic compounds. Very crudely, this is the light independent reaction from your GCSE equation. So now let's get down to the nitty gritty. Hamid, you're allowed to go and have a cup of tea now. Let's talk about photosystems and how the photosynthetic pigments absorb light. So here is my lovely, I'm quite proud of this, lovely but simple diagram of a photosystem. Photosystems are the place where the pigments are fixed into a complex of proteins and photosynthetic pigments. So they're kind of all bundled together, and they are then fixed again, put into the thylakoid membrane. We're going to go all over all of this in a minute. But the photosystem is where the photosynthetic pigments are, and they're just bundled together with a load of protein as well. Don't worry too much about that. What you do need to know are the photosynthetic pigments involved. Chlorophyll A is physically down the bottom of the photosystem. This is called the primary pigment, or your teachers might call it the reaction center. You can use either term, absolutely fine. In fact, nearly always use either chlorophyll A or primary pigment or reaction center. All kind of mean the same thing. Then we have what are called the secondary pigments or accessory pigments in the sides of the photosystem. These green ones are chlorophyll B, so a slightly different type of chlorophyll, and these I'm labeling as carotenoids. You could also include, there are other secondary photosynthetic pigments, and they include xanthophyll and beta-carotene. I've included these because these as examples would be enough to get you the marks. Okay, so these are secondary pigments or accessory pigments. These accessory pigments, this whole photosystem fundamentally acts like a funnel. It funnels the light down. The secondary pigments are the sides of the funnel, the diagonal sides of the funnel, and they funnel the light energy down to the bottom where that chlorophyll A molecule or the primary pigment or the reaction center is. So all of that light energy is funneled down to chlorophyll A because that's the one that does the real work in the light-dependent reaction. But now I want to show you absorption spectra. So look at the slightly faint lines here, because these are the ignore this multicolored one for the moment. These are the absorption spectra for these different photosynthetic pigments. The light yellow greeny one is chlorophyll A, this green one is chlorophyll B, and the browny orangey one is the carotenoids. You can see that along the bottom here, you've got the full rainbow spectrum of light. Richard of York gave battle in vain. All right, because white light is made up of all of these colors. And when I say white light, I am talking lamp light and sunlight. We are getting white light from the sun and from our electric lamps. And that white light contains the full spectrum of colors in it. Some of those colors, those colors are dependent on the wavelengths of the light. So red is a very long wavelength, 700 nanometers. Very short or relatively short wavelength is down at the violet end, 400 nanometers. And you can see that each of the pigments absorb slightly different wavelengths or colors of light. It's one of the advantages of having many different pigments in these photosystems because it means a wide range of wavelengths can be absorbed. And you'll also notice that there is very little absorption in the middle of visible light, i.e. where kind of green and yellow are. And that's because those wavelengths of light aren't absorbed by the leaf or by the chlorophyll. they are reflected off the leaf, reflected off the chlorophyll pigments and these photosynthetic pigments. And those wavelengths reflect off the leaf and into our eyes, and that is why we see leaves as green. And there you go, there's the slight irony. Leaves are green, but it's the one wavelength that they don't use for photosynthesis. You may have known all of that already, so apologies if that's a little bit of revision. This last line here, this rainbow coloured line, is what's called, and I should have called it, the action spectrum, because it is just one spectrum. And this is, as it were, the overall photosynthetic rate at each wavelength of light, taking kind of the average, as it were, of the absorption spectras of all the different pigments. So this is the photosynthetic rate at each wavelength of light. So hopefully that makes that clear. Some of you absolutely need to understand those graphs. So let's have a look at how photosystems work. Light is absorbed by the secondary pigments, chlorophyll B, carotenoids. And this light energy causes electrons in these pigments to get excited. They go up, if you remember your GCSE chemistry, they go up energy levels. But they return back to the pigment that they came from. The electron is excited, but it returns back to the pigment. But what happens is when that electron is excited and then returns to the pigment, it passes on the light energy to the neighboring pigment. So if this pigment has the electron getting excited and then returning to the pigment, it will pass the light energy on to the carotenoid pigment, and the carotenoid pigment will pass it down to the chlorophyll B pigment down here. And this is how the transfer of energy, notice it's not the transfer of electrons, it's the transfer of energy, passes down these secondary pigments, and they are funneled in, the energy is funneled in to chlorophyll A, the primary pigment or the reaction center. So notice energy is passed from pigment to pigment, not electrons, and it goes down to chlorophyll A. Now, you need to be aware that there are actually two different types of photosystem, and they are photosystem 2 and photosystem 1. I'm doing them in that order because that's the order that you are going to come across them when we discuss the light-dependent reaction of photosynthesis. So photosystem 2 is often called P700 because that is the optimum, that is the wavelength of light that the chlorophyll A in photosystem 2 has its maximum absorbance. And equally, photosystem 1 is sometimes called P60 because it's 680 nanometers, which is the wavelength of light which has the maximum absorbance of light for the chlorophyll A in photosystem 1. Excuse me. Okay, so we are about to get down to the light-dependent reaction proper. Remember that list of things that the light-dependent reaction has to produce. ATP, NADPH, so that those can go on to the light-independent reaction, and oxygen. Keep an eye out for those things written in blue on this diagram. So let's just hope you recognize what this is, hoping that you're realizing that this is the double membrane of the chloroplast. Therefore, outside here is the cytoplasm of a cell. And here, inside here, is the single membrane that makes up a thylakoid. So hopefully you're recognizing already, come on, return. This is photosystem 2, the The first one we're going to hit, as it were. This is, now what is going on? Come on. Hold on, just trying to get, this is photosystem 1. And these 3 proteins between photosystem 2 and photosystem 1 are the electron transport chain. They are a specialized set of proteins that can transfer electrons down the electron's energy level. I'll go into that in a second. This at the end is ATP synthase. You can see from its shape why it's sometimes called stalked particles. It has a very distinctive shape. And this is the enzyme. Notice it's ASE, it's an enzyme. And this is what is going to catalyze the reaction. ADP plus PI goes to form ATP. Oh, have I got that? Shri, am I having a moment? Have I got that the wrong way round? Oh, I'm going to have to check. Sorry, guys. Oh, Shri, thank you so much. Yeah, I've got that the wrong way round. Huge apologies. That is really irritating. I obviously wrote it in the slide and just didn't think. This, let me be clear, this is P680 and this is P700. Let's just clarify that. Apologies. Yeah, Julian, when I say a double membrane, this has got 2, you're absolutely right, 2 pairs of phospholipids. There's a phospholipid bilayer here, you know, basically this, yeah, phospholipid bilayer here, and then another one here. So 4 layers of phospholipids in total. That is often not appreciated by students, so delighted that you asked that. Shri, thank you again for picking me up on that mistake. Okay, where are we? What are we talking about now? Yes, so we are dealing with first, we are going to deal with first non-cyclic photophosphorylation. So the making of ATP non-cyclically, in other words, kind of in a linear form. So we are going to have light energy hitting photosystem 2. P680. And as I have already described, and I'm not going to go through it again, these accessory pigments funnel the light energy down into the chlorophyll A. This causes the chlorophyll A, an electron in the chlorophyll A, to be very excited. So excited that it leaves the chlorophyll A. Here's a thought. That means that the chlorophyll A is oxidized, because oxidation is loss of electrons. Just a thought for you. So the electron physically leaves the molecule. And here am I drawing this kind of ever-decreasing bouncing down the electron transport chain to indicate that the electron is lowering its energy level at each stage. And because energy is released at each stage as it goes down the electron transport chain, that energy is used to pump hydrogen ions across the thylakoid membrane into the thylakoid itself. Just so that you know, I'm obviously going through this verbally. In the last slide, I'm actually going to have notes comparing non-cyclic and cyclic. So we'll kind of do a little summary of it again at the end. So we have these hydrogen ions now building up in the thylakoid. I'm sure you don't quite understand why the proteins in the electron transport chain in the progressively lower energy levels. I think I'm just going to say that that is the way that they work because, sorry, excuse the banging that's going on. My daughter is doing some DIY. Because as the electron passes down these protons, it allows the electron to jump down energy levels, and therefore that energy can be harnessed to pump hydrogen ions across the thylakoid membrane. This arrangement, you might already know, also happens at the back end of respiration in oxidative phosphorylation. So here we go. The start of photosynthesis is reflected in the end of respiration because electrons go down electron transport chains in both of them to make ATP. I hope that's all right, Shri. I think that's probably the best I can do. I know this is really weird to get your head around. It's a very weird concept, this whole dropping down of energy levels. I think I'm just going to say, for the moment, I'm going to say go with it. So the energy that is released as the electrons go down the electron transport chain, pump hydrogen ions across the membrane into the thylakoid space. This means that we are getting a buildup of hydrogen ions in this thylakoid space because they can only go back down into the stroma via ATP synthase. And this hydrogen, this hydrogen ion or proton gradient is exactly like water being held behind a dam. It is a form of potential energy for the cell and a way by which something can happen. And in this case, it is the fact that as the hydrogen ions go through this specialized enzyme, and also, let's face it, it's also a transport protein, as the hydrogen ions go through the ATP synthase, they make ATP from, or the enzyme catalyzes the reaction to make ATP from ADP and plus PI. This whole buildup of a hydrogen ion gradient and it going through the ATP synthase is chemiosmosis. Right, where are we going now? And this is photophosphorylation, what we have set out to do by this non-cyclic process. However, non-cyclic photophosphorylation doesn't just do photophosphorylation. It does two other things as well. Because what happens now is that light hits photosystem 1. Come on, return. There we go. Light and is absorbed, just like photosystem 2, light is absorbed by the accessory pigments and the energy is channeled down to the chlorophyll A. And an electron is excited and that it leaves chlorophyll A in photosystem 1. This leaves an electron gap, as it were, in chlorophyll A here. And that is filled by this process here, the electron going down and replacing the electron from chlorophyll A in photosystem 1. So what happens to this electron? Well, now we have the ingredients to make a hydrogen atom. An electron plus a hydrogen ion will make a hydrogen atom, and that hydrogen atom is added to NADP to make NADPH, or reduced NADP. So we have now got two, notice in blue, we have got two things from our light-dependent reaction shopping list. We've got ATP and we've got NADPH, ready to go off to the light-independent reaction. What we haven't done yet is produce oxygen, the last thing on the list. Now, this little bit of information I'm hoping is interesting, and actually I also hope that it helps you link the photolysis of water with photosystem 2, because one of the proteins in photosystem 2 is actually an enzyme that catalyzes the photolysis of water. Photolysis of water means the lysis, the splitting of water with light. And this is what happens. And this, I have done a balanced equation here, because the water is split into 2 electrons, 2 hydrogen ions, and half a molecule of oxygen gas. We're obviously going to accumulate many molecules of oxygen gas as time goes on. But hopefully you can see that the oxygen is going to be used in aerobic respiration, or it's going to diffuse out through the stomata. The hydrogen ions are going to add to this proton gradient that we're setting up in the thylakoid. And these two electrons replace the electrons lost by the chlorophyll A in photosystem 2. Don't worry too much about the fact that I have just had one electron leaving. In fact, if I'm being strictly honest, it will be 2 electrons leaving and 2 replace them. But don't get hung up about the number of electrons. You just need to be able to say that an electron or electrons leave the chlorophyll and they are replaced by the photolysis of water. All of this, I have taught this in a sequence, but all of this is going on at the same time as light strikes the chlorophyll molecules and the other photosynthetic pigments, and electrons are excited. So that is non-cyclic photophosphorylation. Particularly, this kind of electron path is kind of going in a linear flow. And non-cyclic photophosphorylation ticks all the boxes for producing NADPH, ATP, and oxygen, the 3 things that we need from the light-dependent reaction. So now let's go on to talk about the cyclic phosphorylation. So cyclic phosphorylation is only involving photosystem 1. It doesn't involve photosystem 2. Notice that non-cyclic photophosphorylation involves photosystem 1 and 2. So photosystem 1 has light striking it. It absorbs and funnels that light energy down to chlorophyll A, and photoionization happens. But here, photoionization, so the electron is excited, it leaves the chlorophyll molecule, but it goes down part of the electron transport chain. I have actually drawn this accurately. It goes to the middle protein. The electron goes to the middle protein and then drops down the electron transport chain back to the chlorophyll molecule that it came from. So, but as happened previously, as it goes down this electron transport chain, the electron is lowering its energy level, and therefore, as it does so, little packets of energy are released, and they are used to pump hydrogen ions across the thylakoid membrane, as before, into the thylakoid space. And as before, they are channeled through ATP synthase by the process of chemiosmosis to bring about photophosphorylation, the production of ATP. That is where cyclic photophosphorylation ends, because photosystem 2 is not involved. And therefore, we have not got a loose electron coming out of photosystem 1 to combine with the proton, to combine with NADP to make NADPH, and we haven't got the photolysis of water. So the only thing that is made from cyclic photophosphorylation is ATP. When does photosystem 1 do cyclic? So photosystem 1 only does cyclic photophosphorylation in actually, some very simple organisms, so like photosynthesizing single-celled organisms, so they will be eukaryotes because they've obviously got chloroplasts in them, they've got membrane-bound organelles. So, protoctists, photosynthesizing protoctists, may only use cyclic photophosphorylation to produce ATP. Most organisms, most photosynthesizing organisms use both methods of photophosphorylation, and almost predominantly use non-cyclic photophosphorylation so that all the ingredients can be made for the light independent reaction. Light independent reaction. The simplest photosynthetic organisms carry out the rest of photosynthesis by slightly different means. So I hope that makes sense. So I think now we are at the stage where we can actually compare non-cyclic and cyclic photophosphorylation. So hopefully you can see that in both non-cyclic and cyclic, an electron is excited and leaves a chlorophyll A molecule. However, in non-cyclic photophosphorylation, an electron leaves photosystem 2 and it also leaves photosystem 1. In cyclic photophosphorylation, the electron only leaves photosystem 1. I'm hoping that you've got my little diagrams in your mind, so this is kind of making sense. In both situations, the electron passes down, the electron passes down an electron transport chain. In both situations, energy is released that is used to pump hydrogen ions across the thylakoid membrane into the thylakoid. In both situations, a hydrogen ion gradient forms, and therefore the hydrogen ions diffuse through ATP synthase in both cyclic and non-cyclic to produce ATP. So in non-cyclic photophosphorylation, you get loads of ATP because the electrons are going down the whole run of the electron transport chain. Less ATP is produced by cyclic photophosphorylation. These two bits only occur in non-cyclic photophosphorylation. The production of NADPH, because only in non-cyclic photophosphorylation do you have this loose electron, and only photosystem 2 catalyzes the photolysis of water, so only oxygen is produced. So I'm hoping that that gives you a pretty comprehensive but also A-level appropriate summary of those two systems of the light-dependent reaction. And therefore, we have finished cyclic and non-cyclic photophosphorylation. So if there are any questions, really happy to move on and do them. So kind of quick